Hi, this is Curran Simpson. I'm the Chief Technical Officer at Regenxbio. Thank you to the Alliance for Regenerative Medicine for inviting me to do the corporate presentation for Regenxbio. I'll just point you to our forward-looking statement slide. We will be making forward-looking statements in this presentation. If you could look at this slide directly, I'd highly appreciate it. So for those of you that don't know Regenxbio well, we are about 250 FTE company, so a small startup company that's been developing products in the AAV-mediated antibody delivery and rare genetic diseases areas. We have what we call a proprietary NAV technology platform, which primarily encompasses AAV7, AAV8, AAV9, and AAV-RH10. And several of our programs have advanced to different points, I'll show on the next slide, of clinical development, one of which has reached approval via a licensee of ours in the last year and a half or so, called Zolgensma, which has been highly successful. So our pipeline is broken down into three different areas of investigation. Several programs you'll see comprise our retinal disease area, and several programs in our neurodegenerative disease area, treatment of the CNS, and programs also that are liver targeting or muscle targeting as well, all in different stages of development as depicted by this slide. I'll start out with our retinal disease area and describe RGX-314, which has a number of clinical studies either started or well along the way in clinical development. As you know, RGX-314 is intended for the treatment of wet age macular degeneration, which can cause blurring and central vision and progressive vision loss. It's a significant disease affecting hundreds of thousands of patients. And our approach using AAV8 is to generate anti-VEGF antibody fragment similar to what a patient would experience with, let's say, a monthly or bi-monthly injection. In our case, the AAV8 allows the retinal cells to produce the anti-VEGF Fab, and one course of treatment has been shown in clinical studies to give a durable effect on vision, stabilizing vision, and in some cases even improving. I'll talk a little bit about the modes of delivery. So the most advanced clinical studies we have currently ongoing are in the area of subretinal delivery of the vector. And so that's an established route now for delivery of gene therapy. I'll show a little bit of data on that next. And it gives us the most direct and broad transduction of the retina that has been observed. And I think in general, minimal exposure to the vitreous and anterior segment. So the risk of immune response, risk of inflammation is much lower than other modes of administration. We are also exploring, just recently made disclosures around additional programs, and in this case, the delivery method is different. It's supracoroidal, delivering, as you can see from this schematic, to the outside of the eye in the supracoroidal space. This is envisioned to be a potentially in-office, non-surgical approach, and as I said, we are just now working towards starting up our first studies in both wet AMD and also in diabetic retinopathy. So just a little bit more detail on RGX-314 for the subretinal indication. The primary objective was to determine the safety and tolerability of subretinal injection of RGX-314 in subjects with wet AMD through 6 months. And I won't go through all of the secondary objectives, but overall this study was fully enrolled over the last 2 years, and over 42 subjects were administered drug. If you look at this schematic, you'll see that it was a dose escalation study in which there were 5 different cohorts that were conducted at increasing doses along the way. And as this was a first-in-human approach to subretinal administration, we chose this path to fully explore the therapeutic window for this indication and for RGX-314. As you can see on this slide, as measured from aqueous samples by ECL, for each cohort, we measured an increasing level of RGX-314 protein transgene that's expressed. So you see from cohort 1 to cohort 5, a gradual increase and then a bit of a leveling out in the amount of protein expressed after administration of the drug at the levels indicated in GC per eye. And so this is the dose response that we were looking to generate to give us a basis for knowing the transgene expression levels at each cohort. And then for the same set of patients, cohort 1 through 5, we're able to look at the injection rate of prior treatments with other comparator anti-VEGF drugs. And you can see as the amount of protein, transgene protein for RGX-314 is increased through the different cohorts, that the requirement for additional injections for patients dramatically decreases. And you can see between cohort 3, 4, and 5, after 1 year, a substantial reduction, 62%, for example, in cohort 3, and even lower in cohort 5 in terms of less requirement for additional injections. This was monitored also and continues to be monitored for the cohorts out to 2 years, and we see the same level of reduction of additional injections. So the promise here is that people are getting stable protein expression of anti-VEGF and also requiring less frequent injections as a result of that. And in cohorts 3, 4, and 5, stable vision for many of the patients over that course of time as well. So a very good proof of concept, which we intend to advance to additional studies in the near term. So in the other Aviate study that I mentioned earlier, what we'll be exploring is the supracoroidal administration of RGX-314 for wet AMD. And this study is literally being started up as we speak out in the field. And this uses a different device. Again, it's an in-office methodology. The primary study will be safety and tolerability, but we will also measure efficacy by the measures you see on this slide. Interestingly, again, this device is a syringe-based administration expected to be in office, so not a surgical methodology. We're also in the process of starting up a study in diabetic retinopathy, which is the leading cause of vision loss in adults between 24 and 75 years of age. The average age of onset is around 45 to 50 years of age. And so RGX-314 will be administered supracoroidally to these patients, as you see from the diagram below. Again, everything else being the same. AAV8 is the vector. And again, another potential indication for RGX-314 out of wet AMD as our primary indication. We also have this study, it's called Altitude, phase 2 clinical trial scheduled for DR indication. This is just restating some of the information I just gave, but again, the route of administration being supracoroidal in-office treatments. One of the other ocular indications that we're exploring is actually very related to what you'll hear about next when I transfer over to our CNS indications. It's called RGX 381, which is for treatment of ocular manifestations of CLN2 disease. So patients many times will have cellular waste peptides building up, which leads to seizures, vision loss, and neurodegeneration. But with the onset of enzyme replacement therapy, many of those patients now have a better prognosis, better outcome. But having said that, the ocular manifestation, which typically begins at age 4, can lead to rapid vision loss, progressing ultimately to blindness. So in this case, the intended IND that we are working towards utilizing AAV9 will provide a subretinal delivery of TPP1, the same enzyme that is in use for enzyme replacement therapy, which should break down cellular waste within the eye. And our hope is that it will also reduce the manifestations associated with CLN2 disease in ocular indications. Now, switching over to our neurodegenerative disease franchise, we have 3 programs that are active. RGX 121, which is in phase 1/2 for MPS II, RGX 111, which is in clinical development for MPS I, and RGX 181 for CLN2 disease, which is in preclinical development with the expectation to file an IND in the near term. All of these methodologies utilize AAV9 with intracisterna delivery. The most advanced program I'll move to is RGX 121, in which we have completed the enrollment of both of the initial cohorts. So RGX 121 primarily is to determine the safety and tolerability of RGX 121 in severe MPS II patients. And we have many secondary objectives in this study measuring biomarkers related to CSF, serum and urine sampling that is done to look and see if the activity related with MPS II, mucopolysaccharidase 2, is detected and is actually functioning in the CNS space. As I mentioned earlier, both cohorts, dose 1 and dose 2 seen here, are fully enrolled, and these patients are being monitored now. The initial data from the phase 1/2 clinical trial for RGX 121 indicated that it was well tolerated following one-time intracisternal administration, no drug-related SAEs. Patient 1 has completed immunosuppression regimen per protocol, and demonstrated, importantly, consistent and sustained reduction in SF levels of heparan sulfate, a key biomarker of I2S activity. So with this, we see dramatic reductions in some of these storage accumulations in the CNS. Here you see the heparan sulfate change from baseline as measured in CSF. And this gives us hope that this surrogate marker may be leading to positive results in terms of the neurocognitive stability that we're hoping for out of these studies. MPS 1 for RGX 111, we've dosed 1 patient as part of an IIT. And on the next slide, you'll see similar heparan sulfate changes from baseline as measured by in the cerebral spinal fluid. And so again, these surrogate indicators of increased IDUA enzyme activity in CSF indicate to us that we have good expression of the transgene and significant reduction in some of these storage components that need to be broken down to acquire help patients acquire and sustain cognitive developmental skills at a normal rate. I'll switch over a little bit to the NAV Technology platform. We are in the process of building corporate headquarters, which significantly within it will have one story of a GMP facility, which will give us 2,000-liter internal manufacturing capability to enable us to expand our network, which is currently outsourced, with a combination of outsourced and in-source manufacturing. And this is central to many of our programs, the ability to potentially validate and launch our programs from an internal site and improve the robustness of supply through having our own capabilities. We have a very broad NAV technology platform. We have a lot of information acquired over time for AAV7, 8, 9, AAV RH10 in terms of demonstration, not just in preclinical work, but in clinical work to show the advantage of the platform. Many of the programs that I mentioned earlier are also partnerships that have been developed. And you can see the breadth of disease areas that we work in or that our partners work in both, again, liver targeting disease, CNS targeting disease, and also in hemophilia, for example. So we see this as a huge advantage for Regenxbio, is to establish partnerships which enable our programs to move forward through different regulatory paths that have been successful to date. I'll also mention that we have a very experienced team. You can see the names and also the affiliations of many of our executive team with past histories at both large and small biotech, many of which have commercial experience. So as we prepare for late-stage studies in our programs, we look forward to leveraging our executive team to guide us through late-stage development and eventually commercialization. And on our financials, we did disclose, I'll just point out one point here, is $339 million in cash in end of Q2 2020. So we have a strong cash position and interesting milestones near term to continue to fuel our growth and to provide a bright future for Regenxbio. Thank you for your time, and I appreciate, again, the Alliance for Regenerative Medicine for the invite to present our corporate overview.